Welcome to this lesson. In this lesson, we look at transport in animals. Animals are more metabolically active than plants. Because of their greater metabolic activity, they have a greater need for transport of nutrients and oxygen to the tissues and the removal of metabolic wastes from the tissues. In large multicellular animals, the surface area to volume ratio is very small. This means that the volumes of tissues are too large, making the distances traveled by substances to be very large. For these reasons, diffusion alone cannot cater for the transport needs of the organism. As a result, animals have developed circulatory systems for transportation of substances within their bodies. For example, for the transportation of nutrients from the intestines to the tissues where the nutrients are utilized, or transportation of oxygen from the lungs to the tissues, and also transportation of waste products from the tissues to the excretory organs. Now, each circulatory system, regardless of the organism, has the following components. Each circulatory system has a circulating or transporting fluid that is commonly referred to as blood. This is the fluid that carries substances either in solution form or suspension from one part of the body to the other. A second component of the circulatory system are the vessels. Vessels are tubular structures that conduct the circulating fluid. It is through these vessels that circulating fluid moves from one part of the body to the other. Though the extent of the presence of these vessels varies from one organism to another, but every circulatory system should have at least some components of vessels, either partially or entirely. And then the third and very important component is the heart. The heart is a pumping structure or organ that produces the force which pushes the circulating fluid through the circulatory system. Now, there are basically two types of circulatory systems in the animal kingdom. So, namely, open circulatory system and the closed circulatory system. We will start by looking at the open circulatory system. Open circulatory system is one in which the blood is pumped and flows through the body cavity known as coelom. The body cavity of animals is known as coelom. So this blood is pumped by the heart and flows through the coelom. The coelom is the free space between the body wall and organs. So in between the organs in the body, the cavity is known as the coelom, and it is through this coelom that the blood flows. Now, since the coelom is fluid filled, that is, since the coelom is filled with blood, then it is for that reason known as a hemocell. So when the body cavity is filled with blood, then it's known as hemocell. And the fluid, the circulating fluid within the coelom is known as hemolymph. Hemolymph. So in the open circulatory system, let's say that the blood is pumped and flows through the coelom, and this makes the coelom a hemocell. And the blood that flows through the hemocell is known as the hemolymph. Open circulatory system is found in animals such as the arthropods, which include insects, and arachnids such as spiders. Now, if you look at the cross-section of the body of an arthropod, you can see how this open circulatory system looks like. So here, this is the body wall, and then we have the gut. So between the body wall and the gut, that is the alimentary canal, there are organs. The heart and internal organs. The spaces within the body and in between the organs is what is known as the coelom. So it is this fluid-filled coelom that becomes hemocell. So blood is pumped by the heart into the general body cavity, flows between the organs, bringing nutrients and taking away waste products. Then the blood gets back to the heart where it is pumped again, and the circulation continues on and on. The second circulatory system is the closed circulatory system. Now, in this system, the blood is pumped and flows through closed vessels. So here, the blood is confined within closed vessels. It never leaves. The blood never comes into contact with the tissues directly. This system is found in vertebrates, such as fish, amphibians, mammals, and of course reptiles and birds. Now, a cross-section. This is a cross-section of the body of a vertebrate, shows this type of arrangement. There is the body wall and there is the gut. Now, between the body wall and the gut, there are organs. But the blood is not pumped into the body cavity. The body cavity is present, like in all animals, but it is air-filled. It's not fluid-filled. It does not contain any blood. It is air-filled, so it just remains a coelom. But the organs are served directly by blood vessels. So from the heart, the blood is pumped and is conveyed to the different organs through blood vessels. Then from the various organs, the blood is again conveyed by vessels back to the heart. So that is a closed circulatory system. Now we can look at the differences between the open and closed circulatory system. Still tabulated here. In the open circulatory system, blood flows through the body cavity, thus comes into direct contact with the tissues. While in the closed, blood flows through vessels and never comes into contact with the tissues. Secondly, because of the open body cavity, the pressure is rapidly lost after pumping, so the blood flows under relatively lower pressure, and hence there is a lower speed of flow. But in the closed circulatory system, because of the closed and confined blood vessels, once the blood is pumped, the high pressure is maintained. So there is relatively higher pressure that is generated and maintained. Thus, there is a faster speed of blood flow. And because of this higher pressure that is generated, There are two distinct advantages that the closed circulatory system has over open circulatory system, which is one, the blood is pumped over a longer distance since there's a greater force pumping it, so it can travel over a longer distance, and this allows the animals to grow to larger size. Arthropods, such as insects, are partly limited in size because of the open circulatory system that cannot generate enough pressure to cover a long distance. But in the vertebrates, in the closed system, a relatively higher pressure is generated, and this enables the organism to grow to a relatively large body size. As you know, in nature, in many instances, size matters. The larger the animal, the better it will be to fend itself against predators and other enemies, and it will also, the easier it will be for it to obtain its food. Now, the second advantage is the faster rate of blood flow. The higher pressure enables the blood to flow at a faster rate. This means that nutrients are supplied at a faster rate and waste materials are removed faster. This makes the animals to be more active than those with open circulatory system in insects, and in particular using the cockroach. We will look at the cockroach as an example of an insect that has an open circulatory system. Now, in the cockroach, the heart is tubular and is located just above the alimentary canal. So this is a tubular heart, and in fact the only blood vessel that is present in the insect circulatory system is this dorsal aorta at the anterior end. So the tubular heart has 13 chambers. 3 chambers are located in the thorax, while 10 are located in the abdominal segments. You see, the only blood vessel that is present in the cockroach's circulatory system is the dorsal aorta. The dorsal aorta empties into the head regions of the insect. Now, each chamber contains a pair of valves at the anterior end, at this anterior end, that prevent backflow of blood, because blood is propelled forward through the heart by waves of contractions which start from the posterior end and move toward the anterior end. So this propulsion from the posterior end pushes the blood towards the anterior end and out through the dorsal aorta. In the thorax, there are branches of the vessel that take blood to the wings. So blood circulates through the open circulatory system, most of it coming out through the dorsal aorta and into the body cavity. The blood circulates through the legs, into the legs and out, and eventually the blood returns to the tubular heart and enters the heart through openings. Now, each chamber has a pair of lateral opening known as ostium. Ostium is one opening, and plural is they're known as ostia. So after circulation, the blood is sucked into the tubular heart through these lateral openings known as ostia, and then through waves of contractions that begin from the posterior end, blood is pumped towards the anterior, some blood branching out to the wings, most coming out through the dorsal aorta into the head region and back through the thorax, abdomen, and then sucked back into the ostia. So in this way, the insect is able to transport substances from different parts of the body. So for example, as the blood flows or comes into contact with the alimentary canal, it picks nutrients and takes them to the rest of the body. This flowing blood also will come into contact with the excretory structures, where it passes the waste materials to the excretory structures that will then remove them from the body. Now, the circulatory system in the cockroach and arthropods in general has three specific functions. One is the transport of nutrients from the alimentary canal to the body tissues. Also, the transport of metabolic wastes from the tissues to the excretory organs. And third, it provides defense against infections, protects the body against pathogens that may invade the body. This is brought about by the presence of white blood cells. However, the hemolymph, that is the insect's blood, is not involved in the transportation of respiratory gases. It does not transport carbon dioxide and oxygen because the insect's blood lacks pigments, specific pigments that are involved in the transportation of respiratory gases. So unlike other animals, the circulatory system is not involved in the transport of gases. It's mainly involved in the transport of nutrients and respiratory system. You will recall that in closed circulatory system, the blood is confined in blood vessels and never comes into direct contact with the tissues. So the heart pumps blood into the blood vessels, then through the blood vessels, the blood is conveyed to the tissues. Now, there are three types of blood vessels that are involved in the closed circulatory system. The first are the arteries. The arteries convey blood from the heart to the tissues. These arteries divide into smaller vessels called capillaries. Capillaries spread out in a network fashion within the tissues, and are the ones that bring the blood very close to the cells. It is in the capillaries where exchange of substances between the blood and the cells takes place, even though there is no direct contact. So materials must either diffuse across or filter across from the tissues into the blood, and also from the blood into the tissues. The capillaries then eventually reunite to form the third type of vessel known as veins. The veins then take blood back to the heart. Now, there are two types of closed circulatory systems. These are namely single circulation and double circulation. In single circulation, the blood passes through the heart once for every complete circulation. So if this is the heart, the blood is pumped out of the heart, goes through a full circulation in the body, and then back to the heart. That's one complete cycle. In this type of circulation, in the single circulation, the heart has two chambers. The atrium that receives the blood and the ventricle that pumps the blood out. So there is one atrium and one ventricle. So in the single circulation, the heart is two-chambered. This type of circulation is found in fish. So in the fish, the heart pumps the blood first to the gills, where oxygenation takes place. So the blue color here represents deoxygenated blood that is taken to the gills for oxygenation, and once they become oxygenated, they become red. So the red color here represents oxygenated blood. So this here blood has been oxygenated, and from the heart to the gills, the blood is said to be deoxygenated. So the blood then flows through the body tissues where oxygen is taken up, then the blood becomes deoxygenated, flows back to the heart where it is pumped out again. So in single circulation, the blood flows through the heart once, only once, during a complete circulation. Now, the main problem with this type of circulation is that pressure falls considerably as blood flows through the gills, such that by the time the blood gets to the body tissues, it is under very low pressure. So this tends to make the animals, that is the fish, to be less active, or such that they cannot maintain vigorous activity for a long period of time because of the fact that the blood flows through their body tissues at a relatively slow pace. So that means that the rate at which nutrients are brought to the tissues and waste products carried away is rather slow. So that really limits their activity. So in single circulation, remember, the heart is two-chambered, and the blood flows through the heart once for every complete circulation. Now, in other vertebrates, these are different types of circulation known as double circulation. In double circulation, the heart is four-chambered, and the heart flows through the heart twice for every complete circulation. The heart is typically divided into two: the right side and the left side. The right side carries deoxygenated blood, while the left side carries oxygenated blood. So for every complete circuit, the blood will flow through the heart once. So there are two circulations. In the first circulation, known as pulmonary circulation, the blood is pumped from the heart to the lungs for oxygenation, and then it flows back to the heart. That is pulmonary circulation. In the second circulation, the blood is pumped out of the heart to the rest of the body, where it provides nutrients and oxygen, and then it flows back to the heart. This second circulation is known as systemic circulation. Now, the major advantage of double circulatory system is that oxygenated blood flowing through the body tissue is under high pressure. And this therefore means that there's more efficient supply of oxygen and nutrients, thus ensuring high respiratory rates. This makes the animals that have the double circulatory system to be very active, much more active compared to fish. Animals that have double circulatory system include birds and mammals. Some reptiles also do have a double circulatory system. Now, the other advantage is that the deoxygenated blood on the right side is separated from the oxygenated blood. So that means that there is full oxygenation of the blood. And this, as I mentioned earlier, it improves the efficiency of blood or oxygen supply to the tissues. And also, the fact that the blood is pumped twice means that the high pressure is maintained. Because in the first pumping, the blood flows through the pulmonary circulation, and then in the second pumping, the blood flows through the systemic circulation. So this maintains a relatively high pressure, which ensures fast rate of flow, and therefore a more efficient delivery of nutrients and removal of waste materials. In this lesson, we look at the mammalian circulatory system as a representation of the double circulation of vertebrates. Now, in this diagram here, you can see the entire circulatory system of the mammal, consisting of a heart that is connected to various blood vessels that convey the blood to the various parts of the body. The heart consists of 4 chambers. The upper chambers are known as auricles. So we have the left and the right. So this is the right auricle and this is the left auricle. Then the lower chambers are known as ventricles. Likewise, there is a right ventricles, and the left ventricle. Now, oxygenated blood from the lungs. Here are the lungs. So oxygenated blood from the lungs enters the left auricle through the pulmonary vein and then into the left ventricle. That is the left ventricle. So the blood enters then into the left ventricle. Then the left ventricle pumps the blood out of the heart through the major artery known as the aorta. The aorta divides into carotid and subclavian arteries that take blood to the head and upper arm region. So you can see the head and the upper arm regions are supplied by the subclavian artery. Carotid artery takes the blood to the head. Now, the main arteries divide and subdivide into arterioles, that is small arteries, which split into capillaries. So each major organ has many capillaries, through which materials are brought to the tissues, Now, the capillaries are very fine and form a network which is in close contact with almost all the cells of the body. So if you look at all the major parts of the body, there are capillaries, be it the liver, even in the alimentary canal, in the lungs, there are capillaries through which exchange of materials between the blood and the tissues take place. Oxygen and food materials are taken up by the cells, while carbon dioxide and waste products diffuse into the blood at the capillaries. The capillaries then merge to form venules. These are tiny veins, which then merge further to form the main veins that carry the blood out of the respective organs. So each major organ and region has an artery, like for example the kidney has renal artery that brings blood to the organ and renal vein that carries blood away from it. The veins return blood to the right auricle. So from different parts of the body, all the veins return their blood to the right auricle part of the heart. Now, the neck and lower part of the body, it is the blood coming from the neck and the lower part of the body, drain their blood into the inferior vena cava. That is the blood coming from the liver through the hepatic vein, coming from the kidney through the renal vein, coming from the trunk and the legs, through the iliac vein. All these drain into the inferior vena cava, while the blood coming from the head and the neck region drain into the superior vena cava, all of which join and drain the blood into the right atrium. From the right atrium, the blood is pumped into the right ventricle, which then pumps blood out through the pulmonary artery to the lungs for oxygenation. So you can see that if you start from the left auricle, the blood will be pumped out through the general circulation, also known as the systemic circulation, and back to the heart. Then from the heart is pumped to the lungs for oxygenation, and then back to the heart once more, such that for every complete circulation, the blood will have to flow through the heart twice, thus making it double circulation. Now, one important thing to note is that blood brought to the alimentary canal by the mesenteric artery, does not go back to the general circulation directly. Instead, this blood is first taken to the liver through the hepatic portal vein. Now, why is that important? This is because the liver plays a very important role in the metabolism, storage, and distribution of nutrients to other parts of the body. The liver is also the center where detoxification takes place. So anything that may have been absorbed that is harmful to the cells is neutralized within the liver cells. So it's very important that absorbed materials are first taken to the liver for regulation. For example, Any excess glucose is converted into glycogen for storage so as not to raise the blood osmotic pressure too much. Excess amino acids are also deaminated so as not to affect the blood composition. Then from the liver, the blood is then taken back to the general circulation through the hepatic vein, which joins the vena cava, and eventually back to the heart is a muscular organ that is located in the chest cavity. It is located in between the two lungs in this thoracic cavity, and the primary function of the heart is to pump blood to the whole body. Now, in order to understand how the heart is adapted to this function of pumping blood, we look at the structural features, starting with the external structures. Starting with the external structure. In this diagram here, we can see the external features of the mammalian heart. The heart is enclosed in a translucent membrane, though it's not visible in this diagram, known as the pericardium. Translucent membrane known as the pericardium. It encloses the heart. This pericardial membrane secretes the pericardial fluid. Pericardial fluid that acts as a lubricant when the heart is working. The pericardial membrane also helps to keep the heart in position and check on overdilation. It prevents the heart from overdilating. So it checks on overdilation so that the heart does not stretch too much. On the outer part of the membrane, there is fatty tissue. This fatty tissue acts as a shock absorber. Now, the heart is made up of a special type of muscles known as the cardiac muscles. These muscles are specialized, for they contain interconnected fibers. The muscles also can beat, they can contract and relax continuously without getting fatigued. Now, the muscles are supplied with oxygen and nutrients through the coronary artery, which appear on the surface of the heart. Now, the heart has many vessels connected to it, and all the vessels either enter or leave at the anterior end. There's a pulmonary artery that conveys the blood to the lungs. The pulmonary artery branches into the right and the left. The right conveying blood to the right lung, the left conveys blood to the left lung. The aorta is the largest artery that carries blood from the heart. It then branches, and each branch takes blood to different parts of the body. But the main part of the aorta runs on the dorsal side of the heart towards the lower part of the body, hence known as the dorsal aorta. So after branching, after coming from the heart, after exiting from the heart, it passes behind the heart. There's some branches, like the carotid artery, the subclavian artery also branch, taking blood to the neck and the upper limbs and also the head. But most of the blood flows through the dorsal aorta to the rest of the body. There are pulmonary arteries and veins. You have pulmonary vein, which bring blood from the lungs. So pulmonary artery take blood to the lungs for oxygenation, and then the pulmonary veins bring the oxygenated blood back to the heart. We also have the vena cava, anterior or also known as superior vena cava, that bring blood from the neck and the head region, and the posterior vena cava, also known as the inferior vena cava, that brings blood from the chest and below and the lower parts of the body. So on the surface you can see that the heart has two sides. There is the right side, so if you can draw a line here. There is the right side, because the heart is facing us. So what is to your left, that is actually the right side of the heart, because it's facing you. And then the left side, there are 4 chambers, 2 on each side. The upper chamber is known as the atrium, right atrium and left atrium. Then right ventricle and the left ventricle. So in order to understand how these chambers function and how they are structured, we have to look at the internal structure of the heart. The heart is a muscular organ that is located in the chest cavity. It is located in between the 2 lungs in this thoracic cavity. And the primary function of the heart is to pump blood to the whole body. Now, in order to understand how the heart is adapted to this function of pumping blood, we look at the structural features, starting with the external structures. Starting with the external structure. In this diagram here, we can see the external features of the mammalian heart. The heart is enclosed in a translucent membrane, though it's not visible in this diagram, known as the pericardium. Translucent membrane known as the pericardium. It encloses the heart. This pericardial membrane secretes the pericardial fluid. Pericardial fluid that acts as a lubricant when the heart is working. The pericardial membrane also helps to keep the heart in position and check on overdilation. It prevents the heart from overdilating. So it checks on overdilation so that the heart does not stretch too much. On the outer part of the membrane, there is fatty tissue. This fatty tissue acts as a shock absorber. Now the heart is made up of a special type of muscles known as the cardiac muscles. These muscles are specialized, for they contain interconnected fibers. The muscles also can beat, they can contract and relax continuously without getting fatigued. Now, the muscles are supplied with oxygen and nutrients through the coronary artery. which appear on the surface of the heart. Now, the heart has many vessels connected to it, and all the vessels either enter or leave at the anterior end. There is a pulmonary artery that conveys the blood to the lungs. The pulmonary artery branches into the right and the left. The right conveying blood to the right lung, the left conveys blood to the left lung. The aorta is the largest artery that carries blood from the heart. It then branches, and each branch takes blood to different parts of the body. But the main part of the aorta runs on the dorsal side of the heart towards the lower part of the body, hence known as the dorsal aorta. So after branching, after coming from the heart, after exiting from the heart, it passes behind the heart. There's some branches, like the carotid artery, the subclavian artery also branch, taking blood to the neck and the upper limbs and also the head. But most of the blood flows through the dorsal aorta to the rest of the body. There are pulmonary arteries and veins. We have pulmonary vein, which bring blood from the lungs. So pulmonary artery take blood to the lungs for oxygenation, and then the pulmonary veins bring the oxygenated blood back to the heart. We also have the vena cava, anterior or also known as superior vena cava, that bring blood from the neck and the head region, and the posterior vena cava, also known as the inferior vena cava, that brings blood from the chest and below and the lower parts of the body. So on the surface you can see that the heart has two sides. There is the right side, so if you can draw a line here. There is the right side, because the heart is facing us. So what is to your left, that is actually the right side of the heart, because it's facing you. And then the left side, there are 4 chambers, 2 on each side. The upper chamber is known as the atrium, right atrium and left atrium. Then right ventricle and the left ventricle. So in order to understand how these chambers function and how they are structured, we have to look at the internal structure of the mammalian heart. The mammalian heart is composed of 4 chambers. There are 2 auricles, also known as atria, that form the upper chambers, and 2 ventricles that form the lower chambers. The atria have a thin wall and they are smaller in volume. That is, you have the left atrium and the right atrium that are relatively small compared to the ventricles that form the lower part. And also, you can see that the atria have thinner walls compared to the ventricles. The right atrium receives deoxygenated blood from the body organs, except the lungs, while the left atrium receives oxygenated blood from the lungs through the pulmonary veins. The ventricles are composed of thick cardiac muscles. You can see the left ventricle has a thicker wall compared to the right ventricle. The volume of the left ventricle is also smaller than that of the right ventricle. I will see the reason for that when it comes to looking at the pumping mechanism of the heart. Now, there's a thick muscular wall called the Intraventricular Septum that separates the two ventricles. That is, that separates the left ventricle from the right ventricle. This is the Intraventricular Septum. Now, between the atrium and the ventricle, there are atrioventricular valves, also known as cuspid valves. So here, between the right atrium and the right ventricle, there is tricuspid valve, whereas between the left atrium and the left ventricle, there is bicuspid valve. These cuspid valves prevent the blood from flowing back into the atria when the ventricle's muscles contract. The cuspid valves are supported by tendinous cords or valve tendons, which are attached to the walls of the ventricles. They attach to the walls of the ventricles on each side. These tendons prevent the cuspid valves from turning inside out under pressure as the ventricles contract. So that when the ventricles contract, they prevent these flaps of the cuspid valve and the flaps of the tricuspid valves from turning inside out. But the valves on the right side are known as tricuspid because they have 3 flaps. The only 2 are shown here. Whereas the bicuspid valves are so called because they have 2 flaps. But both of them are supported by the tendinous cords that prevent them from turning inside out when the ventricles contract. Now, at the base of the pulmonary artery, here, and at the base of the aorta, here, this is the aorta. So at the base of the aorta and the base of the pulmonary artery, there are cup-like valves known as semilunar valves. So you have the pulmonary artery semilunar valve and the aortic semilunar valves at the base of these 2 blood vessels. These valves are opened by the force of the blood generated by the contraction of the ventricles. The valves also prevent backward flow of blood into the ventricles when the ventricles do relax. Now, circulation of blood through the heart occurs in this manner. Deoxygenated blood from the body tissues, except the lungs, enters the right atrium through the vena cava. Remember, there is a superior vena cava that brings the blood from the neck and the head region, and then the inferior vena cava that brings the blood from the lower part of the body. The blood then flows into the right ventricle via or through the tricuspid valve. When the right ventricle is full, it contracts and pumps the blood through the semilunar valves into the pulmonary arteries to the lungs for oxygenation. The blood from the lungs then flows into the left atrium through the pulmonary veins. So once the blood has been oxygenated in the lungs, then it will flow into the left atrium through the pulmonary veins. Then from the left atrium, the blood flows into the left ventricle through the bicuspid valves. When the left ventricle is full with the oxygenated blood, it contracts and pumps the blood out of the heart into the aorta through the aortic semilunar valve. So when the left ventricles contract, the blood flows into the aorta and to the rest of the body. So through the aorta, the blood is transported to the rest of the body. So the blood passes through the heart twice to complete circulation. First, it enters the heart through the right atrium, then into the right ventricle, then out through the pulmonary arteries into the lungs for oxygenation. Then from the lungs, the oxygenated blood flows back to the heart through the pulmonary veins into the left atrium. Then from the left atrium through the bicuspid valves into the left ventricle. Then the left ventricle pumps the blood out into the general circulation or systemic circulation through the mechanism of the heart. Now, the heart is a special organ in that it has special muscles. The cardiac muscles are special in a number of ways. Now, first, structurally, the cardiac muscle is said to be striated. When viewed under the microscope, each muscle fiber or muscle cell, like this is one muscle cell, has a nucleus, and there appears to be lines or bands, dark and light bands that cut across the length of the muscle fiber. For that reason, the cardiac muscle is said to be striated. Now, when the heart is removed from the body of an animal and placed in a medium that contains the nutrients necessary for its functioning, it will continue to beat for several hours. Now, how is that? This is because the cardiac muscles are specialized in the sense that they initiate their own contractions. That is, the contractions originate from the muscle cells themselves. For this reason, the cardiac muscles are said to be myogenic. That is, their contractions originate from the organ itself. For a muscle to contract, there must be a wave of electric impulse. Now, the other muscles in the body are said to be neurogenic. Neurogenic, meaning that their contractions are initiated from the nervous system. But this is not the case for the cardiac muscles. The cardiac muscles are myogenic. They initiate their own contractions. Another special feature of the cardiac muscle is that it can contract and relax without fatigue. In fact, the moment the heart beats for the first time, in the embryonic stage of an animal, it will continue beating throughout the lifetime of that organism. Cardiac muscles relax, contract throughout one's life without fatigue. Another special feature is the presence of the interconnected fibers. Look at this muscle fiber branches, and these branches enable this line of muscle fibers to be connected to this other line of the muscle fiber. So throughout the wall of the heart, muscle fibers are interconnected, and this is important because it facilitates rapid spread of electrical impulse that brings about contraction. So these interconnections are very important because they help to spread rapidly the waves of contraction throughout the mass of the cardiac muscle. Another special feature are the intercalated discs. The intercalated discs consist of membranes that help to rapidly transmit the electrical impulse from one muscle cell to the next. This way, the spread of contraction is very fast from the point of origin to the rest of the cardiac muscle mass. So the cardiac muscle is highly specialized to function throughout. Even when it is not connected to the nervous system, it will still continue to function. Now, next look at the contractions and relaxations of the cardiac muscles and how these help to bring about the pumping of blood through the circulatory system. Now, the heart undergoes contractions known as systole and relaxation known as diastole. When the ventricular muscles relax, like during diastole, these are the ventricles. So when the ventricular muscles relax, the volume inside will increase. That's the volume increases. Now, when the volume increases, the pressure decreases. At the same time, the cuspid valves, it is a bicuspid valves and the tricuspid valves, open to allow deoxygenated blood from the right atrium into the right ventricle, and also the oxygenated blood from the left atrium into the left ventricle. So during diastole, the ventricle's muscles relax, increasing the volume, thereby decreasing the pressure. This allows for the blood to flow into the ventricles. Now during this time, the atria walls slightly contract to help in the blood flow into the ventricles. So these walls here will slightly contract. Remember, they're not so muscular, they're not so thick, but the slight contraction is important because it helps in the pushing of the blood through the cuspid valves into the relaxed ventricles. So that is what happens during a diastole, relaxation of the ventricles. Now, during the systole, the ventricular muscles contract, and at the same time, the cuspid valves close. The cuspid valves close when the ventricular muscles contract. This will prevent the backflow of blood from the ventricles back into the atria. Remember, when the ventricles muscles contract, the volume decreases. And when the volume decreases, the pressure increases. It is that increase in pressure that pumps the blood from the ventricles and out of the heart. Now, during the systole, the cuspid valves close to prevent the backflow into the atria as the volume decreases or the pressure has increased. At the same time, the semilunar valves of the pulmonary artery and the semilunar valves of the aorta open. So during systole, and as a result of the contractions of the ventricular muscles, blood is pumped from the right ventricle through the pulmonary artery to the lungs for oxygenation. And at the same time, from the left ventricles, blood is pumped out through the aorta into the systemic circulation. If you look at the thickness of the left ventricle compared to that of the right ventricular wall, you see that the wall of the left ventricle is thicker. This generates high pressure, which forces blood to cover the longer distance over the systemic circulation. Whereas the right ventricle generates just enough power to pump the blood through the shorter systemic circulation. So that's why there's a difference in the thickness of the walls. This one is thinner because it pumps blood over a relatively shorter pulmonary circulation, while this, the left ventricle, is thicker because it pumps the blood over the relatively longer systemic circulatory system. So during systole, the ventricle's walls contract to pump the blood out of the heart. A systole is always followed by the diastole, and the two make a complete heartbeat. So heartbeat begins with the diastole into the systole, then back to the diastole, or can also start from the systole, diastole, then back to the systole. So the moment the cycle is completed, then you have a heartbeat. Now, heartbeats can be felt as a pulse or pulsation by placing a finger on the large arteries in the body, like just around the wrist. You can feel the pulsation. Now that pulsation is due to the pushing of the blood. As the blood is pumped through the arteries slightly expand. It is that expansion that is felt as a pulsation. So usually the heart contracts at an average of about 60, 70, 75 times per minute, depending on the age, temperature, activities, and other factors. So the pumping mechanism, you said, occurs alternately. We have rhythmical contraction and relaxation of the heart that occurs throughout an animal's life. Now, let us look at a little bit more about the heartbeat. They say heartbeat is a pulsation, the throbbing action of the heart. And on average, there are about, say, 70 heartbeats per minute. Now, within the walls of the right atrium, there is a group of specialized cells that make up the sinoatrial node. The sinoatrial node serves as the pacemaker that regulates the pace at which the rest of the heart beats. this sinoatrial node generates the electrical impulses that brings about the contraction. It is one that initiates the contraction of the cardiac muscles. So, wave of electrical excitations or impulses originate from the sinoatrial node and spread over the atria. Once the electrical impulses have spread over the walls of the atria, the atrial muscle fibers contract. Then these electrical impulses then collect at another region. There is the junction between the ventricles and the atria, where there's another group of specialized muscle fibers known as the atrioventricular node. The atrioventricular node serves to collect the electrical impulses and boost them, then channels them down the interventricular septum. Along the interventricular septum, there is another group of modified cardiac muscles known as the Purkinje fibers. Now, the Purkinje fibers conduct the nerve impulses along the septum. So the nerve impulses move along the septum to the base of the heart, and then the Purkinje fibers also extend and spread upward like that. So, in this way, the electrical impulses are spread in a very coordinated manner to ensure that the contractions begin at the atria, then are channeled through the interventricular node and Purkinje tissues to the base of the ventricles, and then will spread up. This ensures that the auricles or the atria first contract to pump blood into the ventricles. Then the ventricles contract from the base upward, thereby pumping blood out from the right ventricle to the pulmonary artery, and from the left ventricle and into the aorta. So, the electrical impulses that are generated by the sinoatrial node are transmitted first over the atria, and then via the atrioventricular node and Purkinje fibers to the ventricles. This ensures a smooth, one-directional flow of blood. Now, even though the sinoatrial node does control the rate at which the heartbeat will take place, that's why it's known as the pacemaker. Even when the heart is removed from an organism, it will still continue to beat at its normal, natural rate. That is because the sinoatrial node is one that generates the electrical impulses. And in fact, it is the presence of the sinoatrial node and its function that makes the heart or the cardiac muscles to be myogenic. So their myogenic characteristic is due to the presence of the sinoatrial node, which is a group of specialized muscle cells that generate the nerve impulse responsible for contractions of the muscles. Now, however, the heart can still be controlled and influenced by the brain. As you are aware, under certain conditions, it's necessary to either speed up the rate of heartbeat or slow down. This is brought about by nervous stimulation of the heart. So innervation of the heart simply refers to the stimulation of the heart by nerves. The heartbeat can still be regulated by the brain. There is the vagus nerve and the sympathetic nerves that are connected to the sinoatrial node. The vagus nerve supplies impulses to the sinoatrial node, which serve to slow down the heartbeat. Like when one is asleep, the rate of heartbeat is slowed down, and this stimulation comes from the brain. On the other hand, when there is need to increase the heartbeat, like when one is engaged in vigorous physical activity, then impulses will be sent from the brain through the sympathetic nerve to the sinoatrial node, which then serves to increase the rate of the heartbeat. In this way, the circulatory system is able to adjust to the increased need for nutrients by the muscles and increased need to remove the metabolic waste that is produced at a faster rate during vigorous physical activity. Now, the rate of the heart can also be influenced by certain hormones such as adrenaline. Adrenaline can also speed up the rate of heartbeat. Normally, the adrenaline is produced under certain conditions, like when one is frightened or is exposed to danger. So adrenaline will increase the rate of the heartbeat to prepare the body for vigorous activities such as escape or to fight and overcome the danger that has been presented. Structure and functions of the blood vessels. The main blood vessels are arteries, veins, and capillaries. Arteries transport blood from the heart, and each artery divides to form smaller blood vessels known as arterioles. So from the heart, there's a main aorta that divides into various arteries taking blood to different parts of the body, and then each artery divides into arterioles. The arterioles divide further to form the capillaries. Then capillaries join to form venules, and several venules merge into a main vein through which blood is taken back to the heart. So arteries convey blood from the heart, divide into arterioles, then arterioles into several capillaries, then capillaries join to form the venule, and then several venules merge into a vein, and then all the veins from different parts of the body join the major vein, that is the vena cava, and through the vena cava, blood is taken back to the heart. Now, we first start by looking at the structure and function of an artery. The internal structure of an artery reveals that the wall is made up of 3 layers. There is the innermost layer of artery made up of the endothelium tissue that's known as tunica interna. The endothelium is composed of a single layer of cells. Now, this layer is found in all blood vessels, not just the artery. It provides a smooth lining which offers the least possible frictional resistance to blood flow. Then the middle layer of the artery is composed of elastic fibers and smooth muscles. It's known as the tunica media. This layer is the one that is responsible for the dilation and constriction functions of the blood vessel. The outermost layer, known known as the tunica externa, consist of connective tissues, which are mainly the collagen and elastin fibers. Next, look at the capillaries. Structurally, capillaries are the simplest part of the blood circulatory system. Even though functionally they form one of the most important parts of the circulatory system, because it is at the capillaries where exchange of substances between the tissues and the blood takes place. Now, each capillary is made up of endothelial cells that are only one cell thick. That is the wall. If you look at the cross-section of a capillary, the wall is made up of just one layer of cells. This provides the least distance for exchange of substances between the blood and the surrounding tissues. Now, capillaries are numerous and are very close to the tissues in such a way that each cell is near a blood capillary. This is important in promoting efficient and rapid exchange between the blood and the surrounding cells. Then the veins. Structurally, the veins are very similar to the arteries in that the walls are made up of three layers. There is the innermost endothelium, the tunica interna, the tunica media made up of smooth muscles and elastic fibers, and the tunica externa made up of connective tissue. However, there are some structural differences between the veins and the arteries, owing to the fact that they carry blood that is under different pressure. One of the very distinct difference is that in major veins, there are semilunar valves that project from the endothelium. So the inner lining of the veins do contain semilunar valves at regular intervals. These semilunar valves prevent backflow of blood because the blood flows under relatively low pressure in the veins. So they need to have the semilunar valves to prevent the backflow of blood. Now, the forward flow of blood in the veins is assisted by contraction of skeletal muscles surrounding these veins. So, in addition to the presence of the semilunar valves, the skeletal muscles surrounding the veins also do assist in pushing of the blood towards the heart. Now, functionally, all veins carry deoxygenated blood with fewer nutrients but more nitrogenous waste and other metabolic wastes. Now, except the pulmonary vein that carries oxygenated blood, all the others do carry deoxygenated blood. Now, we can look at the differences between arteries and veins. For convenience, we can look at functional differences and also the structural differences. Functionally, the arteries carries blood that is under considerably high pressure, while veins carry blood that is under low pressure. So the first point of functional difference is the blood pressure. High pressure in the artery, low pressure in the vein. Secondly, And because of the first point, in the vein arteries, the blood flows rapidly and in pulses. Blood flows rapidly and in pulses. But in the veins, blood flows smoothly. So there are no pulsations. And 3, another functional difference is that in the arteries, the blood that is transported is oxygenated. The arteries transport oxygenated blood except the pulmonary artery, whereas the veins transport deoxygenated blood except the pulmonary vein, which transports oxygenated blood from the lungs to the heart. Structurally, if you look at the walls of the two, compare the thickness. So arteries walls are thicker and more muscular than the veins. That is a very distinct difference. Thicker walls in the arteries, thinner walls in the vein. This is because of the thicker layer of smooth muscles. Secondly, arteries have narrower lumen compared to that of the vein. Now remember, the veins transport blood back to the heart, and the blood flowing through the vein is under considerable low pressure. So the wide lumen offers minimum resistance to flow. The arteries have a thick wall to withstand the high blood pressure that flows within it. And also the narrow lumen helps to maintain this high pressure. So these structural differences are directly related to their function. Arteries carry blood under considerably high pressure, hence they need the thick wall to withstand the high pressure, the narrow lumen to maintain the high pressure. In the vein, the blood pressure is considerably low, so the wide lumen offers minimum resistance to flow. Now, a third point of difference is the presence of valves in the veins and the absence in the arteries, except at the base of the large arteries leaving the heart. So that is, except for the outer and pulmonary artery that have semilunar valves at the bases, arteries have no valves. Veins, on the other hand, have valves at regular intervals through their length. Again, this is related to their function. They transport blood back to the heart, which is under considerably low pressure. And so the valves help to maintain the forward flow, that is the unidirectional flow of blood from the tissues and toward the heart. So those are the main differences, both functional and structural differences between the arteries and the vein. Now we can look at tissue fluid formation. Let's look at the process that goes on at the capillaries as the blood flows through them. Now, as mentioned earlier, the capillaries provide the site of exchange of substances between the tissues and the blood, and therefore it is important that they are structured and they are able to facilitate this exchange process. Now, the exchange of substances occurs by a process known as ultrafiltration. Ultrafiltration comes about when the blood flowing through the arterial, which is under high pressure, enters the relatively narrower capillaries. So because of the reduction in the volume, because of the narrowness of the capillaries, pressure builds up. This pressure then forces components of the blood that are small enough to filter out through the capillary walls and into the intercellular spaces. Okay? So the branching of arterioles into capillaries not only increases the surface area for the capillaries and also increases the pressure of blood in them because of the narrowing of the capillaries. And this is what forces molecules that are small enough out of the blood across the capillary walls that, remember, are only one cell thick, and they also do have pores. So the small molecules are forced out into the intercellular spaces. So the molecules that move out and fill the intercellular spaces make up the tissue fluid. This tissue fluid formed is very similar to the plasma of the blood in terms of composition, except that it lacks red blood cells. It does have some white blood cells, and it also lacks large plasma proteins that cannot filter across. However, the tissue fluid contains important substances such as glucose, oxygen, amino acids, vitamins, hormones, and mineral ions that are taken up by the cells either through diffusion or active transport. Now, as a result of the ultrafiltration, there's a drop in the pressure of the blood that remains in the capillaries, such that at this end, the venule end of the capillary bed, the blood pressure is less than that of the tissue fluid, because as the continuous ultrafiltration process takes place and more tissue fluid is formed, the pressure within the tissue fluid will be higher than the reduced pressure at the venule end. So some substances in the tissue fluid will then move back into the blood capillaries at the venule end, and these may include waste substances such as carbon dioxide and nitrogenous wastes. Water will also move back into the blood circulatory system by osmosis, because the filtering out of solutes such as glucose and amino acids raises osmotic pressure of the remaining blood. So even water will move by osmosis back at the venule end. Now, excess tissue fluid that cannot seep back at the venule end enters the lymph capillaries that make up the lymph vessel. So the excess tissue fluid enters the lymph vessel and becomes the lymph. And through the lymph vessel, the lymph is taken back to the circulatory system and rejoins the blood. Then the heart pumps the blood again out through the arteries. Arteries divide into arterioles, and then at the capillary bed there is ultrafiltration. With ultrafiltration, nutrients filter out, they are taken up by the cells. The cells release waste products into the tissue fluid. Some of the tissue fluid seeps back into at the venule end and thus rejoin the blood circulatory system, while some of the tissue fluid will filter back into the lymph vessel, which makes up the lymphatic system, and through the lymph vessels, the lymph is taken back to the blood circulatory system, and the process continues on and on. Defects of the circulatory system. There are a number of diseases these that are acquired due to the environmental factors, and defects. These are structural deformities that may be present from birth that affect the circulatory system. Now we look at three commonest of such diseases and defects. The first of such a disease is thrombosis. Now the term thrombosis refers to the formation of a clot, blood clot, that is, within the blood vessels. There are various factors that can lead to the formation of a clot. One of them is presence of obstructive structures such as cholesterol plaque within the walls of the blood vessel, or could be exposure or breakage of the wall of the blood vessel can also lead to the formation of the clot. But the one that commonly leads to thrombosis, and therefore the disease and defect of the circulatory system, is the presence of structural factors that obstruct the smooth flow of the blood. So whenever we have such obstructive structures, then blood clots. So this is our blood clot. So clot is simply due to the coagulation of blood cells. So thrombosis is formation of a clot within the blood vessel. Now, depending on the part of the body or the organ in which the thrombosis occur, there are different types of thrombosis, and the commonest are coronary thrombosis, and as the term suggests, coronary thrombosis is one that occurs within the blood vessels of the heart, and in particular within the coronary artery, which supplies blood to the heart. So here, this diagram, you can see this, these, the ones in red, these are the coronary artery. Blue represent the coronary vein. So the coronary artery supplies blood and therefore nutrients to the cardiac muscle cells. When a plaque is formed on the walls of the coronary artery, this causes blockage. Now, this blockage becomes increasingly fibrous, or accumulation of fatty materials on the artery also increases. This leads to narrowing of the coronary artery, resulting in less and less blood flowing to the heart. So blood cannot pass freely through the part to the cardiac muscles. Now, depending on where it occurs, if the narrowing occurs here, then a very large part will be affected. If it occurs at this point, then the parts beyond that point are going to be affected. So insufficient oxygen, therefore, will reach these heart muscles that requires a lot of oxygen to function. Now, a serious blockage of the artery can result into a fatal heart attack. So in a heart attack, the cardiac muscles suffocate. The cardiac muscle cells cannot get enough oxygen, and again, depending on the portion of the heart that is affected, the heart attack may be mild or may be massive, depending on the muscle fibers, the mass of the muscle fibers that are deprived of oxygen. Now, coronary thrombosis may be caused by heavy intake of fat, which results to high amount of cholesterol in the blood. As cholesterol accumulates in the coronary arteries, it leads to blockage of the blood vessels, thus causing less oxygen to reach the heart muscles. Other causes may be heavy intake of alcoholic drinks, smoking, being overweight, and psychological and emotional stress. Therefore, to avoid coronary thrombosis, it is important that individuals practice healthy lifestyles by reducing the amount of alcoholic drinks intake, stop smoking, and generally avoiding psychological and emotional stress, not to mention exercising regularly to keep the body weight in check. A second common defect of the circulatory system is arteriosclerosis. Arteriosclerosis refers to the thickening and hardening of the walls of the blood vessels. The hardening of the blood vessels results in permanent damage, because as the blood vessel walls become thickened, they lose their elasticity. And this loss of elasticity will interfere with the normal functioning. Now, the hardening and thickening may be due to deposition of calcium salts or even cholesterol. Now, whenever the inner walls, that is the endothelium, is obstructed, remember the endothelium has a smooth lining to allow the free flow of blood. So whenever this wall is covered with deposition, then this interferes with the free flow of blood. So this deposition may be due to calcium deposit or even cholesterol. Now, the most common type of arteriosclerosis is characterized by excessive growth of fibrous connective tissue in the wall of the arteries. So as more and more materials are deposited, there's growth of connective tissue that is not as elastic. Now this will cause the hardening of the wall. The wall, particularly in the arteries, lose their elasticity. And with the loss of elasticity, then this will increase the resistance to blood flow, leading to overworking of the heart. So as the lumen becomes narrower and narrower, there will be other complications such as heart failure that may result from arteriosclerosis. Now, the causes of arteriosclerosis varies, and this includes being overweight, lack of exercise, emotional stress, and also excessive intake of salts that leads to deposition of calcium on the inner linings of the arteries. Also, consumption of fatty diets that is rich in cholesterol can also contribute. So one way of controlling arteriosclerosis, of course, is to physically exercise regularly, avoiding stress, reducing intake of salt, and taking drugs that reduce the cholesterol levels in the blood. A third defect of the circulatory system is the presence of varicose veins. Now, varicose veins, the condition in which the superficial veins, that is the veins that are just under the skin, become swollen and flabby. This comes about as a result of the failure of the semilunar valves to function. When the semilunar valves fail to function, then they cannot prevent a backflow. You see, in the varicose veins are characterized by being swollen and flabby, so they appear on the surface, especially at the back of the legs, like in this diagram here. In a normal person, the veins should not normally be that visible on the surface. But when the semilunar valves fail to function properly, then they cannot prevent a backflow. So, when there is a backflow of blood, remember, the function of the veins is to transport blood back to the heart. So, a backflow will lead to an accumulation of blood, resulting in a bulge at the point where the semilunar valves, the defective semilunar valves, are present. So it is this collection of blood that leads to the swelling, and the swelling then appears on the surface as varicose veins. In a normal functioning semilunar valves, the valves will prevent a backflow so that there is this continuous smooth flow from the tissues to the heart, and the veins will not swell and thus will not appear on the surface as varicose veins. Now, this disease can be controlled by regular physical exercise of the body and also avoiding being seated for long periods of time, or standing for long periods of time, because the immobility of the legs does place a heavy burden on the semilunar valves, such that when somebody stands for a long period of time with the legs being below the body and the blood has to be moved up against gravity for long periods of time, then this can lead to damage of the semilunar valve. So regular movement of the legs does help in the flowing back of blood through the veins, because remember, the muscles, skeletal muscles around the veins do assist in the movement of blood through the veins. So by exercising regularly physically, these muscles contract and help to push the blood. Otherwise, without the exercise, then the valves may in the long term be overwhelmed with this task of preventing the backflow of blood, especially in the legs, because of the position of the body, the legs being below the heart, and therefore the blood must move against gravity as it flows back to the heart. So regular physical exercise is one way of preventing the development of the varicose veins. Now, a fourth and very common disorder of the circulatory system is high blood pressure, also known as hypertension. Now, the normal blood pressure varies between 90 to 140 millimeters of mercury, that is the systolic pressure, over between 60 to 90 millimeters of mercury, that is the diastolic pressure. That is when the ventricles contract, this is a range of blood pressure that is generated, and when the ventricles relax, the range of blood pressure that is present in the circulatory system is between 60 to 90 millimeters of mercury. Now, any value above 140 over a value above 190 is regarded to be high blood pressure. That is, during systolic pressure, when the value is beyond 140, and when during the diastolic pressure the value is beyond 90, then that is a condition of high blood pressure. Now, high blood pressure is a disorder that is associated with a number of factors, mainly lifestyle factors such as heavy drinking, smoking, taking of large quantities of salt in the food, and general body stress. Now, other disorders such as arteriosclerosis may also contribute to the development of high blood pressure, because arteriosclerosis leads to the narrowing of the lumen of arteries. So this will force the heart to pump more powerfully, thereby increasing the blood pressure. Now, the heart of a hypertensive person usually has to overwork, especially where the cause is the arteriosclerosis. Now, there are a number of complications that can arise from hypertension. One of them is brain stroke. Now, hypertension may lead to the bursting of arteries and capillaries. And should this happen in the brain, the bursting of capillaries leads to leakage of blood, and when brain cells come into contact with the blood, they die, and that's what constitutes a brain stroke. So depending on the part of the brain that is affected, brain stroke and the death of the brain cells may result in paralysis or loss of movement or any other body functions depending on the part of the brain that is affected. In fact, in some cases, the strokes may be fatal if a large part of the brain cells are affected, and if that part controls a vital function, like for example, the heart or breathing. Another complication that arises from hypertension is kidney failure. Again, with the rupturing of capillaries, particularly like in the glomerulus, when these capillaries in the glomeruli rupture, this reduces the functionality of the kidney, and in time the kidney will not be able to efficiently excrete waste products, leading to kidney failure. It can also result in poor visibility. The eyes, especially the retina part of the eye, is very sensitive, and when the blood capillaries there are affected as a result of the high blood pressure, then vision is going to be impaired. They said high blood pressure can also result in arteriosclerosis, because the high pressure may lead to damage of particularly the endothelium. And when the endothelium is damaged, the body responds by depositing or formation of fibrous connective tissue. The fibrous connective tissues narrows the lumen and leads to thickening of the wall, and that's forming arteriosclerosis. Again, with arteriosclerosis, there comes other complications again, such as stroke and rupturing of blood capillaries in other parts of the body. Now, hypertension is more common in individuals that are aged 40 years and above, and it can be controlled through a number of ways that include through regular physical exercises, reducing intake of salt, avoiding excessive drinking of alcoholic beverages, quitting smoking, and also avoiding general body stress by practicing healthy lifestyles and striving to have peace of the mind. Functions of mammalian blood. The mammalian blood consists of a pale yellow colored fluid known as plasma. And in this plasma, there are suspended several types of cells and fragments of cells. The chief function of the blood is to transport oxygen and food materials to the tissues and remove carbon dioxide and other waste materials from the tissues to their sites of excretion. So generally, the following are the functions of mammalian blood. Transport of oxygen from the lungs to the other parts of the body, and also transport of carbon dioxide from the various parts of the body to the lungs. Transport of nutrients from the small intestines to other parts of the body. Transport of waste materials such as urea from the tissues where they are produced to organs where they are excreted. Transport of hormones from endocrine glands to their target organs. Blood is also involved in regulation of body temperature by distributing heat from where the heat is released in organs such as the liver and also skeletal muscles to other parts of the body. Blood also has a defense function against invading pathogens. So, the blood is a specialized tissue, as I mentioned, that is made up of pale yellow fluid known as plasma, and within this plasma there are suspended cells, red blood cells, white blood cells, and cellular fragments known as platelets. We start by looking at the plasma. Plasma consists of 90% water. In fact, 90% of the plasma is water, and the remaining 10% is made up of dissolved substances. Now, about 70% of the dissolved substances are plasma proteins, such as albumins, globulins, and fibrinogen. So the remaining 30% comprises of dissolved organic food materials such as sugars, mainly glucose, amino acids, inorganic salts, and also waste materials such as urea. So in terms of volume, the plasma constitutes about 55% of the blood. Then the remaining comprises of, that is 45% is red blood cell, and only about 1% is made up of white blood cells and the platelets. So within the blood, you have the plasma, in which the other blood components are suspended. Plasma serves the following functions of blood. 1. Transport of nutrients from the small intestines to the other parts of the body. These nutrients are in the form of amino acids and sugars that are transported in soluble form. 2. Transport of waste materials from the tissues where they are produced to the organs, such as the kidney, where they are excreted. 3. Plasma also transports hormones from the glands that secrete them. For example, from the pituitary gland, where ADH is produced, to the kidneys, where the antidiuretic hormone, that is the ADH, functions. So, plasma is involved in the transport of hormones from the glands that secrete them to their target organs. And 4, plasma, being mainly water, is involved in the regulation of body temperature by transporting heat from the highly active organs, such as the liver, to the rest of the body. Next, look at the red blood cells. It's the most numerous cells that make up the blood. So, red blood cells, also known as erythrocytes. Red blood cells are very small cells, about 8 micrometers in length. So at the longest part, they're about 8 micrometers in length, and the thickness at the longest part of about 2.5 micrometers. They are sunk at the center. They're sunk at the center, and this gives the red blood cells a biconcave disc shape. So the red blood cells are characteristically biconcave in shape. In a healthy adult human being, there are about 5 million red blood cells per 1 cubic centimeters of blood. So they are numerous. 5 million red blood cells in 1 cubic centimeters of blood. That's a very large number. Now, red blood cells are enclosed by a very thin plasma membrane, and this helps to promote a fast rate of diffusion of gases, especially into and out of the cell. The cells also lack a nucleus. At maturity, they lack a nucleus and many organelles. Instead, the interior is filled with a red pigment known as hemoglobin. This pigment binds and transports oxygen. So this makes the principal function of the red blood cells to be transport of oxygen from the lungs to the other parts of the body. Now, the red blood cells have the following adaptive features that enable them to transport oxygen efficiently. One, the biconcave disc shape, which provides a large surface area for exchange of gases through diffusion. Two, the absence of a nucleus and other organelles, and instead the presence of hemoglobin, which has a high affinity for oxygen. That is, hemoglobin readily combines with oxygen in areas of high oxygen concentration and to form oxyhemoglobin. So in areas of low oxygen concentration, the oxyhemoglobin will break down into hemoglobin and oxygen. Now, as I mentioned, the absence of the nucleus and other internal organelles provide space for more hemoglobin to be filled. That's another adaptive feature. So this lack of a nucleus and other organelles provide space for more hemoglobin to be filled in, and thus increasing the efficiency of oxygen transportation. Even though the absence of the nucleus greatly reduces the lifespan of the red blood cells. They have a very short lifespan of about 120 days. Another adaptive feature is the thin plasma membrane, which allows for rapid diffusion of gases. Now, functions of the red blood cells, as mentioned before, one, the main function is transport of oxygen from the lungs to the tissue. Within the red blood cells, as I mentioned, there is hemoglobin. Hemoglobin combines with oxygen to form oxyhemoglobin. Oxyhemoglobin is bright red in color and is responsible for the bright color of arterial blood. Hemoglobin alone, pure hemoglobin alone, is dark red in color and hence is responsible for the dark color of the venous blood, that is the blood that is found in the veins. So the blood in the vein is dark red because hemoglobin is unbounded. Blood in the arteries is bright red because most of the hemoglobin have been bound to oxygen to form the bright red oxyhemoglobin. So by combining with hemoglobin and forming oxyhemoglobin, the efficiency of transportation of oxygen is greatly enhanced. In fact, 97% of oxygen is transported in the form of oxyhemoglobin and only 3% in solution form. The other function of the red blood cells is the transport of carbon dioxide. Carbon dioxide is formed in the tissues as a result of cellular respiration. Then it diffuses from the tissues into the bloodstream and is transported to the lungs. Now, about 80% of carbon dioxide in the bloodstream enters the red blood cells, where it combines with water to form carbonic acid. So you have carbonic acid that is formed from the combination of carbon dioxide and water. That is a reaction. Now, in this process is catalyzed by an enzyme known as carbonic anhydrase. Carbonic anhydrase. The presence of the carbonic anhydrase greatly speeds up the uploading of carbon dioxide into the red blood cells. Carbonic acid then dissociates into hydrogen carbonate and hydrogen. So you have this dissociation, hydrogen and hydrogen carbonate. The hydrogen ions combine with oxyhemoglobin, forming hemoglobinic acid, thereby facilitating the release of oxygen by oxyhemoglobin, while the hydrogen carbonate ions pass out of the red blood cells and get into the plasma. It is in this form that the carbon dioxide is transported to the lungs. Once in the lungs, where the concentration of carbon dioxide is low, the hydrogen carbonate ions enter the red blood cells, combine with hydrogen ions, forming carbonic acid. Carbonic acid then dissociates into carbon dioxide and water. The carbon dioxide diffuses out of the blood into the lungs and then it is exhaled. So, red blood cells are involved in the transport of carbon dioxide. In fact, about 80% of the carbon dioxide is transported in the form of hydrogen carbonate ions, and this is made possible by the reaction that takes place within the red blood cell, which is catalyzed by carbonic anhydrase. Now, note that about 15% of the carbon dioxide combines with the hemoglobin also to form carbaminohemoglobin. So this is another form in which carbon dioxide is transported in the form of carbaminohemoglobin. Now, in the lungs, the carb-aminohemoglobin also dissociates, release the carbon dioxide, which is then exhaled. Now, next look at the white blood cells. The white blood cells. Now, unlike the red blood cells, white blood cells have a nucleus, but they lack a pigment, they lack hemoglobin. Hence, the cytoplasm is colorless. In terms of numbers, there are much fewer white blood cells than the red blood cells. In fact, for every one white blood cell, there are about 600 red blood cells. However, the number of the white blood cells increases greatly during an infection. White blood cells are formed in the bone marrow of long bones, and like the red blood cells, which are formed in the bone marrow of short bones, like the vertebral column and the ribs. The white blood cells are formed in the marrow of long bones and also in the lymph nodes. Now, there are two types or groups of white blood cells. White blood cells, also known as leukocytes, are grouped into two, namely granulocytes and agranulocytes. Now, granulocytes are so called because they contain within their cytoplasm granules. These granules give them this, appear as dots. So granulocytes are so called because of the presence of granules, protein granules, within their cytoplasm. These granulocytes have amoeboid movement. They are capable of changing their shape to pass through the walls of the capillaries and into infected tissues. In the tissues and the blood, these granulocytes engulf pathogenic microorganisms through the process called phagocytosis. Hence, granulocytes are also known as phagocytes because they do destroy microorganisms and other unwanted materials through phagocytosis. Once ingested, the microorganisms are digested. Now, some white blood cells may die in the course of phagocytosis. So the dead phagocytes, together with the dead microorganisms and damaged tissues, form what is known as pus. There are three types of granulocytes: a neutrophil, a eosinophil, and basophil. That is the neutrophil, eosinophil, and basophil. A granulocytes, on the other hand, lack granules in the cytoplasm, and they tend to have either a rounded nucleus, like in the lymphocyte, or bean-shaped nucleus, like in the monocyte. So the monocyte have bean-shaped nucleus, while the lymphocyte have rounded nucleus. Now, monocytes are produced in the long bone marrow, just like the granulocytes, and they are also phagocytic. That is, they destroy pathogens through phagocytosis. Lymphocytes, on the other hand, are formed in the lymph nodes, and they produce antibodies that protect the body from infection in a number of ways. One, There are those lymphocytes that produce or they act as antitoxins. That is, the antibodies that produce act as antitoxins. These antitoxins produced by the lymphocytes neutralize the toxins or antigens produced by the pathogenic microorganisms. So some forms of antibodies produced by certain types of lymphocytes are known as antitoxins that neutralize toxic materials from pathogens. In other lymphocytes, the antibodies produced are known as agglutinins. Agglutinins cause clumping together or agglutination of microorganisms. This stops the microorganisms from multiplying and eventually they die. In this way, it makes it easier for the phagocytes to ingest the clumped microorganisms. Still, other lymphocytes produce antibodies that are known as lysins. Lysins destroy microorganisms by digesting their cell membranes or walls, thereby rupturing them. Now, once their walls or membranes are punctured, then the cells are destroyed and microorganisms die. Then you also have lymphocytes that produce antibodies that are known as opsonins. Opsonins adhere to the outer surface of microorganisms, thus making them easier for the phagocytes to recognize and ingest them. Opsonins are like markers. So the antibodies are produced when microorganisms invade the body, and they attach themselves onto the invading microorganisms, thereby marking them and making it easier for the phagocytes to identify and digest them. So those are the various functions of the different types of leukocytes, because the function of the leukocytes depend on the type. Granulocytes are phagocytic, just like the monocyte also, and then the lymphocytes produce antibodies, different types of antibodies that together or generally act to destroy microorganisms that cause diseases. Now, the third component, actually the fourth component of the blood, are the platelets, also known as thrombocytes. Platelets, also known as the thrombocytes. Platelets are fragments from large cells in the bone marrow. They are in fact cellular fragments. They have a complex structure and arise from the division or the breakdown of the cytoplasm of giant cells in the bone marrow known as megakaryocytes. So megakaryocytes break down to produce these platelets. So these are the platelets. They are irregular in shape. Platelets don't have a nucleus, but they have several organelles. Their function is to prevent blood loss after damage of blood vessels through the process of blood clotting. So the platelets do this by forming a plug in the wall of the damaged vessel by producing a substance known as thromboplastin. Thromboplastin then initiates the blood clotting process. Now, the blood clotting process is very important as it prevents loss of blood and also prevents the entry of pathogens. So we now look at the blood clotting process. that is triggered off when a blood vessel is damaged. The process of clotting involves a series of complex reactions where soluble blood protein is converted into a mass of tangled threads known as fibrin that stops the bleeding. Now, when the blood vessels are injured, for example here, when this blood vessel is punctured by the pin, the walls of the vessel is damaged. So this damaged wall produces a rough surface that stimulates the platelets to release an enzyme known as thromboplastin. So here we have the damaged blood vessel. This rough surface, as a result of the damage, will stimulate the platelets to release thromboplastin, or even exposure of blood to air stimulates the platelets to release the enzyme thromboplastin. Now, thromboplastin achieves two things. One, it neutralizes the anti-clotting agent known as heparin. Heparin is a protein that is present in the blood and prevents clotting, blood clotting. So under normal circumstances, the blood will not clot, and the clotting process is prevented by the presence of heparin. So thromboplastin neutralizes heparin. Then secondly, the thromboplastin stimulates or activates the inactive prothrombin, it's another protein, to the active form thrombin. Prothrombin is synthesized in the liver and requires the presence of vitamin K. So prothrombin is one of the plasma proteins, ordinarily to not be activated unless there is a presence of thromboplastin. Under normal circumstances, it is the heparin that prevents the activation of the prothrombin. So the release of thromboplastin by the platelets first neutralizes the heparin, and secondly activates the prothrombin into thrombin. This activation process also requires the presence of calcium ions. Prothrombin is made in the liver and it requires vitamin K input. Then it is released into the bloodstream. On exposure, platelets release thromboplastin. Thromboplastin then activates prothrombin to thrombin. And this activation, this conversion requires the presence of calcium ions. The active thrombin then stimulates the conversion of the soluble protein fibrinogen into the insoluble fibrin. The fibrin forms a meshwork of fibers. Forms a meshwork of fibers on the cut surface that trap the red blood cells to form a clot. So this fibrin forms strands that are insoluble on the surface of the damaged blood vessel. So this traps the red blood cells, and the trapped red blood cells therefore will prevent further loss of not only blood cells but even the body fluids. So as the clot dries up, it forms a scab. As it dries up, it forms a scab that stops the bleeding and protects the damaged tissues from infection. So this process of blood clotting is summarized in this diagram here. First, there's the damage to the blood vessel. That damage to the vessel stimulates the platelets release thromboplastin. Thromboplastin then neutralizes heparin and also activates prothrombin into thrombin. Thrombin then activates the conversion of the soluble fibrinogen into the insoluble fibrin. The insoluble fibrin fibers then form a meshwork above the cut surface here to stop further bleeding and also prevent infection by pathogens. Thus, blood clotting reduces loss of body fluids. Now, excessive loss of blood, especially more than 2 liters, can lead to severe anemia unless it is corrected by blood transfusion. Now, it is important to note that in humans there are some individuals that lack the clotting factors, the factors that are necessary for the conversion of prothrombin to thrombin or fibrinogen to fibrin. This condition is known as hemophilia. When any of the factors along the step are absent, so that the process cannot be completed, then you have a condition known as hemophilia. Hemophilia may arise due to genetic disorder or can be due to lack of vitamin K. Individuals that are afflicted with hemophilia may bleed excessively from minor cuts, which can be fatal. Now, injections of vitamins containing vitamins K may be given to prevent excessive bleeding if the cause of hemophilia is lack of vitamin K. Red blood cells have certain proteins called antigens on their plasma membranes. These antigens. Now, these antigens stimulate the immune system to produce specific antibodies. So, an antigen is any substance, usually protein in nature, that stimulates the immune system of an organism to produce antibodies that are specific to it. Now, based on the presence of certain antigens, there are 2 main blood grouping systems, namely the ABO system and the rhesus factor system. In the first system, that is the ABO system, there are 2 types of antigens designated A antigen and B antigen. Blood groups are determined by the types of the antigen present in an individual's red blood cell. Thus, an individual whose red blood cells have A antigen is grouped as blood group A. So, this is a red blood cell. If on the plasma membrane there is antigen A, then that blood is grouped as A, blood group A. Likewise, an individual in whose blood the red blood cells have B antigen, that blood group is B. Then there are those individuals on whose red blood cells antigens A and B are both present. So here you have antigen A, B antigen is present, so that group is designated as AB. While those individuals whose red blood cells lack neither antigen A nor B are said to belong to group O. So basically there are 4 blood groups under the ABO system. Blood group A that has antigen A, blood group B with antigen B, blood group AB that has both antigens A and B, and blood group O that lacks neither A antigen and B antigen. Likewise, the plasma too has antibodies. Antibodies that are present in the plasma which correspond to antigens A and B. There are two types of these antibodies, designated antibody A, small letter A, and antibody B. Antibody A reacts with antigen A, while antibody B reacts with antigen B. Now, if an individual has antigen A, that is blood group A, he or she cannot have antibody A. So naturally, individuals that have blood group A have antibody B in their plasma. Likewise, those people that belong to blood group B, meaning they have antigen B on their red blood cells, have antibody A. While those that belong to blood group AB that have both antigens cannot have either antibody A or B. So they have no antibodies in their plasma. They have neither antibody A nor B. While the blood group O that has no antigens on the red blood cells has both antibody A and antibody B in the plasma. Now, as mentioned, antibody A, for instance, reacts with the antigen A. So if red blood cells that say have antigen A comes into contact with blood or plasma that has antibody A, there's going to be clumping together, or what is known as agglutination. These antibodies will cause the clumping together of the red blood cells that have the corresponding antigen. So it is very important that antibodies and antigen do not correspond, or they do not occur together in the same circulatory system, for there is going to be an antigen-antibody reaction or antibody-antigen reaction resulting in agglutination. And this is what is taken into consideration during blood transfusion. Now, blood transfusion refers to the transfer of blood from a donor into the circulatory system of a recipient. A recipient will receive blood from a donor if the recipient cannot produce antibodies corresponding to the donor's antigens. That is, the transfusion is going to be compatible if the recipient's blood does not have corresponding antibodies to the donor's antigens. So during transfusion, what is taken into account? You look at the donor's antigens vis-a-vis the recipient's antibodies. Because if the donor has, say, antigen A and the recipient has antibody B, then there's going to be an agglutination. There's going to be an antibody-antigen reaction. So during transfusion, we look at the recipient's antibodies. By knowing the recipient's blood group, we can tell the antibodies present, and then we match that to the donor's blood group. If the antibody and the antigens correspond, then you avoid the transfusion and you say that two bloods are incompatible. Now in this table here, summarizes the compatibility of the different types or groups of blood during transfusion. If you take blood group A with antigen A, has antibody B. So if blood group A is the donor, meaning that blood group A can give to another blood group A and can give to AB. Why AB? Remember, AB does not have any antibody, so it will not matter the type of antigen present on A. Whereas blood group A, if he is a recipient, he can receive from another blood group A and blood group O. Because blood group O does not have antibodies that will react with the antigen in his red blood cells. Blood group B has antigen B and antibody A. Therefore, if B is the recipient, he can receive from another B and O, but not A, because the antibody A will react with the donor's A antigen. Blood group B can give to another B, of course, and can also give to AB, because AB does not have antibodies to react with the B antigen. Blood group AB does not have any antibody, meaning that can receive from AB, can receive from A, can receive from B, can receive from O, because it has no antibodies to react with the donor's antigen. For this reason, blood group AB is said or is referred to as universal recipient. Individuals that have blood group AB are said to be universal recipients because they can receive blood from all the other groups, since they don't have antibodies to react with the donor's antigen. Blood group O does not have any antigen, but has both antibody A and B, meaning that O can receive only from O, because it has the two antibodies, and can only receive blood that does not have any antigen, and that is blood group O. However, blood group O can give to itself, can give to A, because it has no antigens to react with the antibody B in blood group A. Can also give to B, since it has no antigen to react with the antibody A in blood group B, and can also give to AB, since it has no antigens to react with any antibodies. But also remember, blood group AB does not have any antibodies, so it can receive from blood group O. Now, because blood group O can donate to all the others, the blood group O is said or is referred to as universal donor. It can give to all the other groups because it lacks antigens. Blood group AB is universal recipient because it lacks antibodies that will react with the donor's antigens, while blood group O is universal donor because it lacks the antigens to react with the recipient's antibodies. Now, in addition to the ABO system, there is another grouping system based on another type of antigen, that is the rhesus factor antigen. So the red blood cells may also have another antigen on their membrane known as the rhesus antigen. Individuals with the rhesus antigen on the membrane of the red blood cells are said to be rhesus positive, while those individuals whose red blood cells lack the rhesus antigen are said to be rhesus negative. Now, on average, about 85% of the population are rhesus positive, and only about 15% are rhesus negative. Now, normally, the rhesus negative person does not have antibodies against the rhesus antigen. Normally, the rhesus negative person does not have antibodies against the rhesus antigen. However, when rhesus positive red blood cells are introduced into the circulatory system of a rhesus negative person, then they will develop the anti-rhesus antibody. So normally during transfusion, to prevent development or formation of anti-rhesus antibodies, rhesus negative persons are not transfused with rhesus positive. So for example, if one is blood group A negative, meaning that they have antigen A, but they lack the rhesus antigen, then they can only receive blood from A negative and O negative. Because if you transfuse this recipient with the A positive blood, then the rhesus factor on the red blood cells will cause the formation of antibodies against the rhesus factor, and that may lead in future to agglutination. So rhesus negative persons receive rhesus negative blood from corresponding groups. If it is B negative, gets B negative or O negative, but not negative recipient getting positive, rhesus positive blood. But on the other hand, these rhesus positive individuals can receive blood from rhesus negative individuals without any harm. Now, when a rhesus negative woman marries a rhesus positive man, the woman is likely to conceive a rhesus positive fetus. So you have the rhesus positive father, rhesus negative mother. This fetus here is most likely to be rhesus positive. Now, the rhesus positive antigen of the fetus may pass across the placenta. May pass across the placenta into the mother's bloodstream during the last month of pregnancy. The mother's bloodstream or the immune system responds by producing rhesus antibodies. So the rhesus antibodies are produced in response to the presence of the rhesus positive red blood cells. Now, the rhesus antibodies destroy some of the red blood cells of the fetus because these antibodies can pass across the placenta back into the fetal circulatory system. So, within the fetal circulatory system, the rhesus positive red blood cells are destroyed by the anti-rhesus antibody. Now, the firstborn child has a higher chance of survival because the destruction of the red blood cells is minimal. However, in the subsequent pregnancy, if the fetuses happen to be positive, then there's going to be massive destruction of the red blood cells leading to serious symptoms and deformities of the baby, a condition known as hemolytic disease of the newborn, or erythroblastosis fetalis. However, to prevent erythroblastosis fetalis, the mother can be treated with anti-rhesus globulin. Anti-rhesus globulin, which prevents her from producing antibodies against the fetal antigens. So that during the last month, she's treated with this anti-rhesus globulin, so that the presence of the rhesus positive red blood cells is not recognized by her immune system. So she will not develop anti-rhesus antibodies. This will protect the red blood cells of the fetus in the present and the subsequent pregnancies from destruction. However, after birth, if the baby is still alive, the baby can be transfused with the rhesus negative blood to remove the destroyed red blood cells and also get rid of the rhesus antibodies in the baby's system, so that the circulatory system can function normally. So whenever a rhesus positive father marries and have a child with a rhesus negative, there are high chances of getting a rhesus positive. Now in the first pregnancy, the baby may not suffer as much, because by the time sufficient antibodies are produced, the baby will have been born. But the subsequent ones are the ones that will suffer, because these antibodies remain. They linger on in the mother's circulatory system and would invade the subsequent fetal circulatory system. Should the baby be rhesus positive, then there will be massive destruction of the red blood cells. Such as the mammals. There is a second transport system that supplements the blood circulatory system. This is the lymphatic system. The lymphatic system and blood circulatory system are functionally and structurally linked, in the sense that in the blood circulatory system, wherever we have blood capillaries, wherever there are blood capillaries, ultrafiltration, the process of ultrafiltration takes place. You call this process ultrafiltration that takes place in the capillary beds. Now, through ultrafiltration, there is formation of tissue fluid. Tissue fluid carries nutrients to the cells. Now, some of the tissue fluid seeps back into the blood circulatory system at the venule end, but the excess drains into lymph capillaries. So not all the tissue fluid can drain back into the venule end of the capillaries, so the excess seeps into the lymphatic capillaries within the tissues. So the lymphatic capillaries join to form larger lymph vessels, which join to form even larger lymph vessels that carry the lymph back to the circulatory system. So, in general, the lymphatic system is made up of lymph vessels that begin at the capillary bed, that is within the tissues. You have blind ending lymphatic capillaries. So, through the lymphatic vessels, the excess tissue fluid that enters it now becomes a lymph and then is transported back to the blood circulatory system, where the lymph from various parts of the body, like these ones here, represent the systemic capillaries, and here the pulmonary capillaries. So wherever you have capillaries, there will be formation of the tissue fluid, and the excess tissue fluid drains into the lymph vessels, becoming the lymph. So the lymph vessel transports the lymph back to the blood circulatory system, where the lymph joins the blood circulatory system at the vena cava. So as soon as the tissue fluid enters the lymph vessel, it becomes the lymph. Now, along the lymphatic vessels, there are lymph nodes that filter the lymph to screen out and destroy any pathogens that may be present in the lymph. Along the length of the lymph vessels, there are valves that prevent a backflow to ensure that the lymph flows in the general direction towards the heart. So, what is a lymph? Lymph is simply the excess tissue fluid that drains into the lymphatic capillaries and then is transported back to the blood circulatory system through the lymph vessels. So the two circulatory systems are related. The two transport systems are related structurally and functionally. As the heart pumps blood out, first to the pulmonary circulation, within the lungs there are lung capillaries, and at the capillaries there will be tissue fluid formation, and the excess tissue drains into the lymphatic capillaries. Likewise, in the rest of the body, the blood is pumped out of the heart through the aorta and carried by the arteries to various parts of the body. In the tissue capillaries, there will be filtration. The excess tissue fluid formed will drain into the lymphatic capillaries, and through the lymph vessels, blood is transported back to the blood circulatory system. Sorry, not the blood, but the lymph is transported back to the blood circulatory system, where it will be joined, where it will drain into the vena cava and thus rejoin the blood circulation. Now, lymph vessels branch throughout the body, and they carry lymph from different parts of the body back towards the heart. This diagram here, you see the network of the lymphatic system. The lymph vessels originate from all the parts of the body, from the arms, the legs, even the head. Now, along the lymph vessels, there are nodes. Now, you note that these nodes are concentrated in particular regions, like under the armpit, around the neck, around the intestines, and the groins. Now, the function of the lymph nodes is to screen and remove pathogens. It is also in the lymph nodes where white blood cells, the leukocytes, are produced. The white blood cells, specifically the lymphocytes, are produced, and these help to protect the body against invasion by pathogens. So the location of these lymph nodes is very important. Those that are located under the armpit serve to protect the body against invasion. Should there be any wound in the arms, an open wound, and pathogens happen to enter through it, these pathogens will be screened out by these lymph nodes under the armpit. Same case, should there be an injury on the head or in the mouth, like when you bite your tongue or your cheek, the open wound is an entry point for pathogens. So these lymph nodes here around the neck screen out these pathogens and prevent them from getting into to the center of the body. Along the alimentary canal, the same, should pathogens enter the body across the walls of the alimentary canal, then they will encounter the various lymph nodes that will screen them and destroy them and prevent them from getting to the center of the body. Around the groin area, again, the lymph nodes perform the same function, whereby should there be an injury in the extremities, such as the lower limbs, that is the legs, then pathogens that happen to be present in the lymph can be screened out and destroyed by the lymph nodes. Response. An immune response is the process of antibody formation as a result of the presence of antigens or pathogens. Recall, an antigen is usually a protein substance that is present on the plasma membrane of a cell. Now, the presence of a foreign antigen stimulates the body to produce antibodies. An antibody is a chemical substance, usually protein, which is formed in the blood when an antigen is introduced into the tissue of an animal. An antibody has a chemical composition which is complementary to the antigen against which it reacts. So each antibody is produced to specifically react with or combine with a particular antigen. So this illustration here shows what happens in an immune response. The introduction of pathogens or any other foreign antigen into the body is recognized by a special type of white blood cell. There's a special type of white blood cell that recognizes foreign antigens. Then the white blood cell stimulates the lymphocytes to multiply and increase in number, and also stimulates the lymphocytes to produce antibodies that are specific to that particular pathogen or antigen that has been recognized. The lymphocytes release the antibodies, which then attack and neutralize or destroy the antigen or the cells that bear the antigen. So basically that is how the immune response comes about. Introduction of a pathogen or a foreign antigen, identification by a particular white blood cell, which then sets in motion the response, in which there is multiplication of lymphocytes. These are white blood cells that produce antibodies, and they produce specific antibodies that will then attack and destroy the specific antigen. So the presence of antibodies for a particular or against a particular antigen brings about an immunity. So immunity is the ability of a body to resist an invasion by pathogens. The ability of the body to resist an attack by pathogen is known as immunity. There are two types of immunity: natural immunity. This is one which is inborn. It is inherited. It is genetically determined, present from birth, and can be passed on from the parent to the offspring. And then there is acquired immunity, one that comes about after birth. It is not present at the beginning, but because of exposure to environmental factors, the organism is able to acquire the immunity. Now, the acquisition can come about either naturally or artificially. Naturally, when a person, for example, is attacked by a particular disease, by natural means, not deliberately, somebody falls sick, so is attacked by diseases like smallpox or chickenpox, measles, mumps, or polio. And when such a person recovers from the attack, then they develop resistance or immunity to any subsequent infections from the same disease. So what happens is that when the body is exposed to the pathogens that cause the disease, white blood cells respond by producing antibodies, and these antibodies fight off the infection, and this is what brings about the recovery. Now, some of the white blood cells that produce specific antibodies remain in the circulatory system for life, so that whenever there is a subsequent infection, whenever the foreign pathogens, the pathogens that cause, for example, chickenpox, should invade the body, then they respond very quickly by producing antibodies to destroy the pathogens. So this is how the immunity is naturally acquired without any deliberate effort on the part of the person. A second way of acquiring immunity is by artificial means. This is through the agency or action of man. Now, artificially acquired immunity can come about in two ways. One, it may be actively acquired, and this is normally done through vaccination. Now, a vaccine is a preparation that contains either dead pathogens or weakened pathogens, or portions of the pathogen cells that contain the active antigen. So the vaccine is then injected into the body, the process known as vaccination. The vaccine will not cause the disease because the pathogen is either dead, is weakened, or is only a portion of it that has been injected. Once in the body, then the dead pathogen is recognized by the white blood cells, which then respond by producing antibodies against it. These antibodies would usually remain in the bloodstream for a long period of time or even for life, depending on the nature of the pathogen, and thereby providing immunity. Now, it is known as actively acquired immunity because the body is stimulated to produce its own antibodies. Vaccine is normally administered either through oral inoculation, through the mouth, or the vaccine can be injected directly into the bloodstream. Either way, the vaccine that contains the dead pathogens will then stimulate the body to produce its own antibodies, which will then protect the body against any invasion by the specific pathogen. Now, a second way of artificially acquiring immunity is by passive means. By passive means. Passive in the sense that the body is not stimulated to produce its own antibodies, but rather antibodies are introduced into the body. Antibodies already formed by another body or another animal are introduced into the body. This occurs in two ways. One, from mother to the fetus during pregnancy. Certain antibodies can pass across the placenta from the mother to the fetus, thereby conferring upon the fetus when it is born some immunity. Secondly, during breastfeeding, especially the first milk, known as colostrum, is rich in antibodies. So the baby is able to acquire these antibodies and get protection or immunity, even though temporary, because antibodies, being proteins, have a limited lifespan. So from mother to the fetus or mother to the baby, antibodies are passed naturally. The other passive way of acquiring immunity artificially is through injection of serum that contains antibodies. Now, serum is plasma from which fibrinogen has been removed. Now, using certain animals such as the horse, specific antiserum can be prepared. The horse may be injected with a pathogen that causes a disease, for example, like rabies. The horse produces antibodies, then the horse's plasma is collected, fibrinogen is removed so that the serum that remains will contain the antibodies against rabies. So this antiserum can be injected into a person to specifically protect them from rabies or even tetanus. So again, this is a passive form of immunity because these antibodies have a limited lifespan and they will be destroyed in due course of time. But the advantage in the injection of antiserum is that it provides immediate protection. It provides immediate immunity. And this is important when during an emergency, for example, if it is a snake bite, what is injected, what is administered is antivenom serum, so as to neutralize the toxins that may be present in the snake's venom. Allergic reactions and organ transplants. What is an allergy? An allergy is a hypersensitive reaction to an antigen by the body. Or another way of putting it, an allergy is a drastic reaction of the body seen in some people towards certain foreign substances or antigens that are normally harmless to the rest of the population. So the body responds to the presence of certain foreign antigens. These antigens may be present in otherwise ordinary substances like pollen grains, certain foods, dust, fur, and so on. So the body responds drastically by overproducing antibodies. So during an allergic reaction, the white blood cell, the lymphocytes that produces antibodies, overproduces antibodies against these otherwise harmless antigens. These antigens that bring about allergic reactions are also known as allergens. So allergens may be found in pollen grains, may be found in some drugs, and so on. So here is the white blood cell. It responds by overproducing antibodies. Now, some of these antibodies remain and adhere onto the white blood cells and also other cells. See, when antibodies are produced, they are released into the plasma where they react with the antigens or antigen-bearing cells. But if they are overproduced, some will remain adhering onto the surface of the white blood cell and other body cells. The result is that the antibody-antigen reactions occurs on the surfaces of the cells, with the result that these cells burst, releasing histamine. Histamine is a chemical substance that increases the permeability of the epithelial cells of the capillaries, thus making them to take in fluid and swell up. The intercellular spaces to become filled with fluid because the histamines will increase the rate of ultrafiltration. So the swelling up of the tissue, the inflammation results in pain. Now these reactions can occur anywhere, not just on the white blood cells, they can also occur on any other cells in the body, resulting in a number of manifestations or conditions of allergy. These allergic reactions or manifestations are many and vary depending on the persons. So they may include, for example, if the reactions occur on the surface of the eye, inflammation of the eye, and reddening of the eye also. Can have stuffy or runny nose, itchy and sore throat. Parts of the body like the lips may get inflamed. The respiratory tract may also get swollen and constricted, leading to asthma, where you have difficulty in breathing as a result of an allergic reaction. So the walls swell and the lumen of the respiratory tract becomes narrower, making breathing quite difficult. There may be development of skin rashes. The eustachian tube, the hollow tube that connects the middle ear to the pharynx, may also get blocked, and this impairs hearing and may actually lead to infections of the middle ear. Cavities in the head, the sinuses may become swollen, and this makes breathing difficult, and the swelling may produce headache. So different people have different allergens that cause their allergic reactions. For some people, it may be certain drugs like penicillin, aspirin, that may cause rashes. Or others may be types of food or certain proteins like groundnuts, eggs, that may cause the itching, sneezing, or even vomiting. In others, dust particles irritate the respiratory tract, bringing about an asthmatic attack. Now, under severe condition, the histamines may cause too much swelling in the body. That is, the histamine may increase the permeability of the epithelial cells to the extent that the blood pressure may drop. Now, lower than normal blood pressure is dangerous and may lead to what is known as anaphylaxis. Anaphylaxis attack, when there is severe drop in the blood pressure. Now, this is how bee stings can cause death in some people. The bee sting contains allergens that increases the permeability of the epithelial cells. A lot of filtration takes place from the blood into the tissue fluid, resulting in swelling and, more importantly, a drop in the blood pressure. If there's severe drop in the blood pressure, this can cause death. So different people respond differently and to different extent to allergic substances, or that is the allergens. Now, one way of avoiding allergic attack is to avoid the allergens. Identify the allergens that cause the reactions, and that way you avoid the reactions. Or through the administration of antihistamine drugs such as Piriton to control the swelling. Then you also have organs transplants. Now, organ transplants has become increasingly important in that in many people, certain organs and tissue may become diseased or malfunctional and may be therefore necessary to replace them. Now, an important aspect of the animal body is that it's able to distinguish between its own cells and those from another individual, regardless of whether they are of the same species or different species. So, when carrying out organ or tissue transplant, it is important to ensure that the antigens present on the donor's organ or tissue will not be rejected by the recipient's body. Just like in blood transfusion, the cells of the organs or tissues to be transplanted have antigens, and these antigens may be identified as foreign and be rejected by the recipient's body. So that is one of the biggest challenges when it comes to organ transplants. Now, it's been possible to transplant a number of organs and tissues from one person to another, or even from an animal to a person. Some of the organs and tissues that are commonly transplanted include the eyes, or just the surface, the cornea, lungs, kidneys, pancreas, even skin. Bone, especially bone marrow, is transplanted, especially where we have a case of leukemia. Tendons, these are the ligaments that attach muscles to bones, can also be transplanted. Veins, portions of the intestines, liver, and of course heart and heart valves. These are organs and tissues that are commonly transplanted after ensuring that the recipient body will not reject the organ. Once that one is done, then the transplant can be carried out. Now, in case there is the urgent need to transplant an organ, even when there's a risk of rejection, then certain drugs can be administered to suppress the immune response of the recipient. These drugs suppress the rejection of the donated organ or tissue. In this way, the organ or tissue that is be transplanted can be kept alive, and thereby the patient will also be kept alive.